How's it going, everyone? Today, I would like to present my senior thesis project titled Neurovascular Oxidative Stress is Associated with Cerebral Amyloid Angiopathy in Alzheimer's Patients via NADPH oxidase and heparan sulfate proteoglycan activity. So what does that mean? Let me break this down in the next few minutes for you guys. So Alzheimer's disease basically currently affects 5.8 million Americans, and this figure is projected to be much higher by 2050. So cerebral amyloid angiopathy is a vascular dementia. It's a vascular form of dementia in which, dementia is a multifactorial disease in which we think, and the NIH also thinks, and has been funding a lot more research into vascular contributions to dementia, which means the blood vessels that lead to the brain and other parts of the central nervous system. So previous work from the Zipfel lab has shown these three separate proteins to be implicated in exacerbating CAA. So HSPG is a cell surface initiator of A beta-induced NADPH oxidase activation, oxidative stress, and vascular dysfunction. And NADPH oxidase is one of the key proteins that produces a lot of vascular oxidative stress, which plays a causal role in CAA pathogenesis. So this VOS basically exacerbates CAA through a lot of different mechanisms, which I won't get into today for sake of time. And also, one of the main ways why this causes CAA is because LRP1 is one of the main clearers of amyloid beta, this big aggregating protein that plays a huge role in dementia. NADPH oxidase will produce a lot of VOS, and this hurts LRP1. LRP1 is a protein that clears away the A beta from the blood-brain barrier. So without LRP1 functioning correctly, we can't clear A beta away, and therefore there's more aggregates in the cerebral vasculature. So a lot of these results have not been translated yet to humans, and so the main goal of my project was to translate these results in vivo rodent models as well as in vitro cell culture models into human patients. And I hypothesized that there will be a correlation between NADPH oxidase levels, HSPGs, and also oxidized LRP1 with amyloid beta expression in the cerebral vasculature. So how do we accomplish this? We first received 10 human brains from both Alzheimer's and also non-Alzheimer's patients as a control from the Knight Center at WashU. I sectioned them and stained them to confirm the presence of CAA in the humans, or if there wasn't CAA also. I also isolated the cerebral vessels. So we're studying here cerebrovascular, right? A vascular disease. So we can't just take the whole brain, crush it up, and then measure the proteins in that liquid, in that protein sample. We have to get the blood vessels first, and then we lyse them and get all the proteins out of the blood vessels. So this includes endothelial cells and also arteries, smooth muscle cells, and just all the cells within the neurovascular unit of these patients. We also had immunoprecipitate LRP1 because it was in too low concentration to increase the total concentration in the protein samples. We use Western blots to quantify the level of proteins. So I basically quantified in all 10 samples amyloid beta, NADPH oxidase, HSPGs, and LRP1s, and just correlated them together to see if they would match up, and kind of be aligned with our hypotheses. So results in figure 2, you can see that yes, there is presence of CAA in these samples. There's hallmark pathology of CAA seen within in figure 2C, where you see these ovals. These ovals are kind of cross-cut sections, almost like imagine like slicing a blood vessel down the middle, and you have the lumen on the inside and the arterial or capillary wall on the outside. Figure 3 shows the overall quantification of amyloid beta. There's not much to kind of really talk about there. We use Western blots to quantify the amyloid beta, and we use beta-actin as a loading control. So this allows us to have a baseline of how much amyloid beta is in each of these samples in their blood vessels. So figure 4 discusses NOX2 GP91. NOX2 is, again, the major catalytic subunit of NADPH oxidase. So having high NOX2 levels in an in vivo model, we would think would lead to greater oxidative stress and more CAA. And do we see that here? We actually do. Let me zoom in a little bit for you guys here. So you can see the amyloid load relative to the control is a positive and significant correlation. The R squared is not as important in these studies, as we're not trying to make a predictive model. Instead, we're trying to test the association. So the p-value of under 0.05 is very promising for this. We also correlated GP90, glucagon-4, a huge major HSPG that is responsible in rodent models for disrupting the neurovascular unit in CAA. And we saw positive correlation between glucagon-4 and amyloid, as well as NOX2 and glucagon-4, which may suggest a synergistic interplay between HSPG, leading to the downstream activation of NADPH oxidase, leading to LRP1 inactivation or inhibition. Same here, syndicin-1 is another HSPG that we are testing here, and the p-value here for the correlation with amyloid, it's a little bit shaky. It's 0.34, but there is still a positive correlation. So hopefully with more samples in the future, we'll be able to correct that and have a statistically significant correlation there. NOX2 GP91 and this HSPG syndicin-1 do show a positive correlation, which is a promising sign again that these proteins are linked in the same pathway. Again, these are just correlative studies, so we're using these correlative studies to suggest, but we can't, confirm a causal pathway until we test it in maybe a clinical trial or additional rodent models. LRP1 results are a little bit unpromising here. One correlated with amyloid load. We measured HNE4 and LRP1. HNE4 is a measure of oxidative stress in these LRP1s. So the greater the HNE4 to LRP1 level, the more oxidized the LRP1 will be. And unfortunately, the correlation was slightly positive, but not significant statistically. So hopefully more samples in the future will take care of that. So overall, we found significant correlations and associations between NOX2 GP91 and CAA pathology, as well as syndicin-1 and glucagon-4 and CAA pathology. And there was a positive non-significant correlation between glucagon-1 and A beta, glucagon-1 versus NOX2 GP91, and syndicin-1 versus A beta. But the correlation between HSPGs and NOX2 supports this hypothesis that yes, these proteins work in tandem to exacerbate CAA formation. And hopefully LRP1 correlation with CAA will be a little bit more significant in the future when we repeat these studies with 25 more brain samples, which is what we're currently doing. So we want to translate ultimately this target of this drug, maybe targeted drug inhibition, and also different in vivo models to NADPH oxidase and HSPGs, and maybe a clinical trial one day to see a broad-reaching effect on vascular dementia approaches. So thank you all. I want to acknowledge the Zipfel lab, Dr. Graham Zipfel, Jogender Mella were two of my main mentors in doing this project, as well as the whole Zipfel lab as a whole. Thank you, everyone. I'll stop recording this. Oh. Nice.